Imagine sinking. The light above you goes from blue to grey to nothing. The pressure doubles, then doubles again. Your ears would pop at 30 meters. By 300 meters, the weight pressing on your body would be 30 times what it is right now. By 1,000 meters, you have crossed the boundary. The sun is gone, not dim, gone. And in that total dark, something is moving toward you. You cannot see its body. You cannot hear it approach. You cannot feel the pressure wave of anything large because nothing is rushing. It is patient. It has been patient for hours. And the only thing it has done, the only thing it does, is pulse a small light in front of its own face. Beautiful, steady, waiting for you to swim toward it. That is just one creature. We will meet dozens more tonight, and they live beneath every ocean on this planet. Right now, while you are listening to this, Chapter 1, The Darkness Below, Why the Deep Creates Monsters. If you enjoy our nighttime science journeys here on Earth at Night, please take a moment to like this video and subscribe to the channel. We love it when our community shares thoughts in the comments and reacts to each other. That is where the real conversation begins. Now let us start. Tonight we are going somewhere most living things have never been. In the hours ahead, you will meet a fish with teeth so large that closing its own jaw would impale its brain unless its skull grew specific holes to receive them. You will encounter a shark whose lineage predates the first dinosaurs by over 200 million years. You will learn about a form of reproduction in which a male dissolves its own body into its mate and the two of them fuse into a single permanent living creature. And you will understand exactly why. The deeper you descend into this ocean, the more extreme and disturbing everything becomes. Every detail is true, and when this journey ends, you will never picture this planet the same way again. Right now, beneath every ocean on this planet, something is happening that almost nobody thinks about. The ocean goes down, not in the shallow, pleasant way you imagine when you picture a beach or a sunlit reef. The ocean descends into a darkness so complete, and a cold so permanent, and a pressure so enormous that the creatures living at its greatest depths have spent their entire evolutionary history on a different planet. A planet that shares a surface with ours. A planet that covers more than 70% of the world. A planet we have mapped less thoroughly than the surface of Mars. The deepest point of this ocean is the Challenger Deep in the Mariana Trench. It sits at approximately 11,000 meters below the surface. If you lowered the tallest mountain on Earth into that trench and stood at its highest point, you would still be submerged more than 2 kilometers down. That is the scale of a world that exists directly beneath us every single day. And through every meter of that descent, something changes. Something gets stranger. The life that exists at 200 meters below the surface looks unusual. At 1,000 meters, it looks wrong. At 4,000 meters, it looks like it should not be possible. And at the very deepest levels, the organisms there look less like animals and more like the products of a completely different idea of what life is and what a body is for. This is not accident. There is a precise and unsettling logic behind every strange feature of every deep-sea creature. That logic lives inside three forces. Three pressures acting on living things over hundreds of millions of years. Three forces that intensify with every meter of descent. Together, they explain why the creatures at each layer of the ocean are always more extreme than those above them. The first force is darkness. The sun pushes light into the ocean as far as light can travel. In clear tropical water, on the best possible day, that reach is roughly 200 meters. But the sun does not deliver its full spectrum even close to that distance. Red light is absorbed within the first 5 to 10 meters of the surface. A wound beneath 10 meters of water produces blood that appears black. Not because anything changed in the blood itself, because there is no red wavelength remaining at that depth to reflect back to your eye. Orange is gone by 20 to 30 meters. Yellow follows shortly after. Even blue, the most persistent wavelength in seawater, fades to absolute nothing, somewhere between 1,000 and 1,200 meters. Below that depth, no photon of light from the sun has ever reached. Not once in 4.5 billion years of planetary history. This is not dim. This is not the darkness of a room with the lights off. This is the permanent, total, and complete absence of all light across an environment that covers close to 1/3 of the surface of this planet. Remove the light, and you remove everything that depended on it. Color serves no purpose where there is nothing to reflect it. Eyes built for sunlight become expensive, organs with no return. Some deep-sea lineages lost their eyes almost entirely over millions of years. Others grew eyes to extraordinary sizes to capture every trace of biological glow in the surrounding water. Both responses are exactly correct. Both are evolution removing what stopped working and amplifying what still does. The deep ocean did not go dark in the way we mean when we say dark. It went dark to the sun. But approximately 75 to 80% of all organisms living below the sunlit zone produce their own light through bioluminescence, a cold chemical reaction that converts energy directly into photons without generating heat. The deep is not empty blackness. It is alive with cold fire. Every creature there is either generating light or hiding from it or hunting with it. The darkness did not end the war between predator and prey. It moved that war into an entirely new dimension. The second force is pressure. One atmosphere presses on everything at sea level. Our bodies were built inside this force, and we do not feel it. At 500 meters depth, the pressure reaches 50 atmospheres. At 1,000 meters, it is 100. At 4,000 meters, it climbs to 400 atmospheres. At the bottom of the Mariana Trench, it exceeds 1,100 atmospheres. That is the weight of approximately 50 commercial aircraft pressing down onto a surface the size of your clenched fist. Bone cannot survive these forces unchanged. Rigid structures compress and fracture. Gas-filled cavities collapse inward. Evolution found its answer across hundreds of millions of years. It made the creatures soft, gelatinous, built from tissue that yields rather than shatters. The deep sea is the domain of transparent flesh and boneless forms. Creatures that look fragile by our standards are precisely engineered for conditions that would destroy anything rigid. In their world, they are exactly right. The third force is starvation. No photosynthesis occurs in the deep ocean. Every creature below the sunlit zone depends on what falls from above. Marine snow, a slow and endless rain of organic particles drifting downward through thousands of meters of dark water, dead plankton, broken fragments of organisms, fecal matter, shed exoskeletons. By the time this material reaches depths of 3,000 meters, it has passed through so many organisms and been so thoroughly degraded that it arrives as nutritional dust, barely enough to sustain life. Scattered across an enormous dark environment with no edges and no floor in sight, deep-sea creatures may go weeks or months between meals. This is the engine that produced the most extreme predatory biology on this planet. When food is rare enough and the intervals between eating are long enough, the body evolves to catch and hold absolutely everything that comes within range. Jaws that open to angles that look physically impossible. Teeth too large to retract. Stomachs that expand to hold prey twice or 3 times the size of the animal itself. Lures, traps, ambushes built on weeks of patience. Every feature of every deep-sea predator that looks grotesque and wrong traces directly back to this brutal equation. Food is rare. Missing it may be fatal. Waste, nothing. Darkness, pressure, starvation. These 3 forces, working together across hundreds of millions of years, have produced the most extreme biology on this planet, and they compound with depth. The deeper you descend, the more absolute the darkness. The heavier the pressure, the rarer the food. The creatures at each successive layer are a little more extreme than those above, a little more shaped by conditions that long ago pushed life past what we thought was possible. Tonight we follow that gradient from the first place where the sun fails to the deepest floors of the world. We will meet the creatures that evolution built for each layer of that descent. We will understand the logic behind everything that looks like a nightmare. And we will discover that the most extreme life on this planet does not live in volcanoes or frozen deserts. It lives directly below the ocean's surface, in a darkness that existed before almost anything alive today was born. Let us go down together. Chapter 2. The First Horrors. Where Twilight Zone creatures begin. 200 meters below the surface, the sun gives up. Not in the way a sunset fades. Not gradually and not gently. The light compresses. What was once the full open sky of light above a diver becomes a pale circle, then a narrow disk of weak blue. Then nothing at all. The world above closes like an eye being slowly shut from the outside. And below that closing, there is a darkness that has no floor in sight and no light source anywhere except the cold chemical glow of the creatures that learned to make it themselves. This is the entry point to the mesopelagic zone, the twilight zone, the region between 200 and 1,000 meters where sunlight is not fully absent but is so vanishingly faint that it cannot power photosynthesis and can barely register on instruments. Scientists describe light at 500 meters as approximately 1 million times dimmer than full daylight at the surface. A single photon landing on a sufficiently evolved retinal cell is worth more than food at these depths. The evolutionary pressure here is unlike anything on the sunlit surface. It is paranoid, it is relentless. And every creature that evolved inside it carries the marks of that pressure somewhere on its body. What makes the twilight zone biologically extreme is not only the fading light. It is the combination of that fading light with the first onset of real food scarcity and the beginning of serious pressure. Animals in this zone are wedged between two worlds, too deep for reliable sunlight, not deep enough to have fully abandoned it. They are caught in a transition zone where evolution is actively tearing apart old solutions and building new ones. The results are bodies that look like nothing on the surface and nothing in the true midnight zone either. They are evolutionary experiments running in real time, and some of them are extraordinarily disturbing. You feel it most immediately in the teeth. The viperfish. Choliodes sloani measures between 25 and 30 centimeters, about the length of a human forearm. It is silvery and narrow, and lined along its flanks with rows of small bioluminescent organs called photophores. None of that is what you notice first. What you notice first are the fangs. The viperfish carries the longest teeth relative to head size of any fish in the twilight zone. The upper jaw fang teeth are so large they cannot be contained inside the skull when the mouth closes. They curve outward past the eyes. In a fully grown specimen, they extend back toward the tail. If you scaled a viperfish to human height while preserving the tooth proportions, you would have fangs the length of your arms projecting from your face. The viperfish does not close its mouth in any useful sense. It simply navigates the world with its enormous teeth arranged around a skull too small to contain them, and it turns those teeth into a weapon of extraordinary violence. The viperfish does not chase prey. It accelerates toward it, directly, at speed. At the last moment, the jaws swing open and the fangs hinge outward. The fish hits its target and impales it. Nothing caught on those teeth retreats, because the curve and length of the fangs make backward movement impossible. The viperfish then walks the prey down its own throat using a hinged jaw mechanism that moves food backward in alternating steps. The whole sequence is less like eating and more like a spring-loaded trap firing. The viperfish also carries a second hunting system. The first ray of its dorsal fin has been modified over millions of years of evolution. It extends forward over the head into a long, flexible spine that ends in a glowing bioluminescent tip. In the dark water of the twilight zone, this light hovers just above the fish like a lantern over a trap door. Small creatures drawn toward any light source approach it. The distance closes. The fangs fire. One animal. Two entirely separate hunting strategies, both born from the same darkness and the same hunger. Now meet the fangtooth. Anoplogaster cornuta rarely exceeds 15 centimeters in length. When researchers first studied juvenile and adult specimens side by side, they concluded they were looking at entirely different species. The juvenile is pale and streamlined with delicate spines, extending from its head like a small crown. The adult is squat and iron dark, with a rough scaled body, and a jaw that belongs on something 10 times larger. The scientific reclassification that united them took years of comparative anatomy. But the real horror of the fangtooth is not its transformation. It is the holes in its skull. The fangtooth carries the largest teeth relative to body size of any fish currently known to science. The lower jaw fangs are so disproportionately large that the fish evolved a structural solution to a problem its own biology created. When the fangtooth closes its mouth, the lower fangs would drive straight into the roof of its skull and pierce its brain. So the skull grew holes. Two deep recessed sockets in the bone of the upper palate, positioned to receive the lower fangs precisely when the jaw comes to rest. Without those sockets, the fangtooth would impale its own brain every time it was not actively hunting. A creature that had to evolve damage control systems inside its own skeleton to survive having its own face. Now, the hatchetfish. Because the hatchetfish solved a completely different problem, and the solution is one of the most elegant in the ocean. The marine hatchetfish belongs to the family Sternoptychidae and is named for the shape of its body. Viewed from the side, it narrows at the back and widens at the front like a blade. Its flanks are coated in guanine crystal mirrors that reflect the surrounding dark water. From the side, the hatchetfish is nearly invisible because its mirrored surface returns the exact darkness of the water behind it. But the real engineering is on its belly. Along the underside of the hatchetfish runs a ridge of photophores, tiny biological light sources in a precise row. These produce blue-green light at exactly the right intensity to match the faint downwelling illumination still reaching down from the surface hundreds of meters above. A predator looking upward would normally see the dark silhouette of a fish blocking the light from above. The hatchetfish eliminates that silhouette by replacing it with light. From below, you see no shadow. You see unbroken faint light, as if the fish were not there. And the system adjusts in real time. If a cloud passes over the sun at the surface, 500 meters above the hatchetfish, dims its belly lights to match the reduced illumination. The photophores are calibrated continuously. The hatchetfish does not hide in darkness. It manufactures invisibility using the exact quality of the light it is trying to vanish from. Before we leave the twilight zone, there is one more creature worth knowing. One that almost never appears in documentaries, but is likely the most numerous vertebrate on this planet. The bristlemouth fish, tiny translucent creatures belonging to the genus Cyclothone and its relatives, 3 to 5 centimeters long, nearly transparent, lined with rows of small photophores along their undersides, unremarkable to look at, but they are everywhere in the twilight zone. Dense clouds of them drift through the water column in aggregations that stretch for hundreds of meters. Scientists estimate they exist in numbers ranging from hundreds of trillions to potentially quadrillions across the world's oceans, the most numerous vertebrate alive, and you have almost certainly never heard of them. This chapter has introduced creatures from a single zone of one ocean, a fish that impales prey on teeth too large for its own skull, a fish that evolved holes in its head to survive its own face, a fish that builds a real-time light-based cloaking system, and the most numerous vertebrate alive that nobody talks about. And we have not yet descended below 1,000 meters. In the next chapter, we go deeper and meet the creatures that turn the simple act of seeing into the most extreme biological engineering on this planet. Chapter 3. The War of Eyes, the optical arms race of the mesopelagic. In a world where sunlight is gone, and the only light is what living creatures generate themselves, the eye becomes the most contested piece of biology in the ocean. On the surface, a functional eye is enormously valuable, but it is not everything. Smell, hearing, vibration, sensing, and speed all contribute to survival. Remove the light, and the equation changes completely. In the deep ocean below the sunlit zone, sight becomes the single most decisive survival advantage or disadvantage any creature can have. If your visual system is sensitive enough to detect a bioluminescent flash 100 meters away, you find food. You avoid predators. You survive. If it is not, you become food for something that can. Over hundreds of millions of years, this single pressure has bent and twisted and broken and rebuilt the eye into forms that have nothing in common with the round, familiar organ you have in your own head. The deep sea produced tubular eyes, mirror eyes, eyes that rotate inside transparent skulls, eyes sensitive to wavelengths of light that no other creature in the same environment can perceive. Eyes so large they dwarf the entire head, and eyes that were simply abandoned altogether when sight became more expensive than it was worth. This chapter is about one of the most extraordinary results of that pressure: a fish with a transparent skull, and what was hiding inside it for decades before anyone thought to look. The barrel eye fish. Macropina microstoma is a small fish of the mesopelagic zone. Adults are rarely more than 15 centimeters long. For most of the 20th century, scientists who studied preserved specimens collected in nets noticed its most obvious feature immediately. The top of its head was a transparent, fluid-filled dome. Through that dome, two bright green glowing structures were clearly visible. Scientists assumed these were the fish's eyes. They appeared to point straight upward through the dome. The fish was catalogued as having two tubular, upward-pointing eyes enclosed in a transparent head shield. And that was the accepted understanding for decades. It was completely wrong. In 2009, researchers from the Monterey Bay Aquarium Research Institute managed to film a living barrel eye in its natural habitat using a remotely operated underwater vehicle. When they reviewed the footage, they saw something that forced them to re-examine everything they thought they knew about this animal. The two bright green structures were not fixed in a permanent upward gaze. They were moving, rotating, swiveling smoothly and precisely from pointing straight upward to pointing directly forward. The eyes could rotate through a full range of positions inside the transparent dome. The fish could look up to spot prey silhouetted against the faint surface light above, then swivel its gaze forward to target and pursue it. The transparent dome that everyone thought was just a structural feature protecting the head was not merely protection, it was a housing. A biological cockpit canopy built to allow full rotation of the most specialized visual organs in the twilight zone. And the dome was not glass clear. It was filled with fluid that filtered incoming light to a specific wavelength, reducing the scattering that would otherwise degrade the image quality of an eye trying to detect a single glowing creature in otherwise black water. What everyone assumed was a simple anatomical quirk turned out to be one of the most sophisticated visual systems in the ocean. And the two dark holes above the barrel eye mouth that look like eyes in every photograph, those are its nostrils. The barrel eye uses its extraordinary eyes to do something precise and calculated. It hovers motionless in the water column beneath siphonophores, those long translucent colonial organisms that trail curtains of stinging tentacles through the water. The barrel eye watches with its upward rotating eyes as small creatures become entangled in the siphonophore tentacles. Then it steals the catch, picking paralyzed prey from the tentacles with surgical precision using its small mouth, the transparent dome protecting the eyes from the stings of the very trap it is raiding. Now meet a fish that took visual evolution in a completely different direction, the telescope fish. gigantura species, named with admirable directness for the obvious feature. Its eyes are not spherical. They are elongated tubes projecting forward from the head, parallel to each other, pointing forward like a pair of binoculars attached to a face. Where most fish have eyes positioned on the sides of the head, giving a wide field of view and limited depth perception, the telescope fish sacrificed peripheral vision entirely in exchange for something much more valuable in its environment. Binocular overlap, the ability to judge distance precisely. In complete darkness, where light comes only from bioluminescent organisms of varying sizes, at varying distances, the ability to judge exactly how far away something is becomes the difference between a successful strike and a missed meal. The telescope fish traded breadth of vision for precision. It evolved its eyes into rangefinders. But the most extraordinary visual specialization in the deep ocean does not belong to any fish with unusual eye shapes. It belongs to the dragonfish. Malacosteus, niger, and Aristostomias titmani are species of lucejawed dragonfish found in the mesopelagic zone. They carry bioluminescent organs that can produce not just blue-green light as most bioluminescent organisms do, but far red and near-infrared light, a wavelength close to the boundary of visible light that most deep-sea organisms simply cannot detect. Here is what that means. In the midnight zone and the deep twilight zone, the visual systems of almost every creature evolved to detect blue and blue-green light. That is the wavelength of bioluminescence. That is the wavelength that penetrates best in seawater. Over hundreds of millions of years, the eyes of deep-sea animals converged on sensitivity to those wavelengths because those are the wavelengths worth seeing. Far-red light was invisible at the surface because the sun delivers almost none of it at depth. No creature evolved to see it. There was nothing to see. The dragonfish changed that equation by producing far-red light itself. It hunts by illuminating prey with a wavelength of light that the prey cannot see. To a shrimp or a small fish in the midnight zone, a dragonfish hunting with its red photophore active is completely invisible. There is no searchlight, there is no warning. The predator is lit up in its own private visual spectrum, while the prey swims in what it perceives as total darkness. The dragonfish is using a channel of the electromagnetic spectrum that it shares with no other creature in its environment. And to detect the reflected red light bouncing back from prey, the dragonfish evolved a retinal pigment derived from chlorophyll, a molecule taken from plant-based photosynthesis chemistry and repurposed for predatory vision in the deep ocean. The dragonfish borrowed a plant's light harvesting tool and converted it into a hunting weapon. In a world where every visual advantage is shared and every predator can see the same bioluminescent tells, as every prey item, the dragonfish found a way to step entirely outside the existing system. It invented a private channel, and then it was the only creature that could receive the signal. The eyes of the deep ocean are not simply bigger or smaller versions of what you know. They are the products of pressures that have no equivalent on the surface. They reflect millions of years of a war where sight was the only weapon that truly mattered, and where every innovation in predation immediately spawned an innovation in evasion. The barreleye built a rotating telescope inside its own skull. The telescope fish fused its eyes into binocular rangefinders. The dragonfish invented a private frequency that its prey will never evolve to detect before it is too late. We have not yet reached the depths where the last traces of ambient light disappear completely. We are still in the zone where creatures need eyes at all. In the next chapter, we leave the war of vision behind and descend to where the only adaptation that matters is the mouth, where the deep ocean produces the most extreme feeding architectures in the history of life. Chapter 4. Mouths from Nightmares. Predatory jaws that defy biology. There is a fish in the deep ocean that is almost entirely mouth. Not mouth as a feature of an otherwise normal body, not a prominent jaw on a recognizable creature. Mouth as the central organizing principle of everything the animal is. The jaw is the largest structure. The stomach behind the jaw is second. The rest of the body, the organs, the narrow whip-like tail, the faint bioluminescent glow at the tail tip. Everything exists in service of what that mouth might one day contain. This chapter descends into the deep ocean's most extreme answer to starvation. And to understand why these creatures exist the way they do, you first need to understand how completely the food situation breaks down at depth. Marine snow begins its long descent from the sunlit surface as relatively nutritious material, dead plankton still containing fats and proteins, fragments of organisms, shed exoskeletons still carrying chemical energy. But the descent takes weeks. And through every meter of it, bacteria and small organisms intercept the falling material and feed from it. Scientists estimate marine snow loses approximately 90% of its caloric content between the surface and a depth of 3,000 meters. By the time it arrives at depth, it is nutritional dust, almost nothing. Barely enough to sustain organisms with the slowest metabolisms on the planet. The predators of the deep cannot wait for dust. They need living tissue, protein and fat and chemical energy stored inside a living body. And living bodies at depth are rare. They arrive at intervals measured in days or weeks. They do not announce themselves. They pass in the dark without warning and without returning. A creature that misses the opportunity may not encounter another for a very long time. You might think a reasonable solution would be to evolve patience, and deep-sea predators did evolve patience of extraordinary degree. But patience alone does not catch a meal. What the deep ocean also produced. Across many separate lineages over hundreds of millions of years was the mouth, the body of the gulper eel, Eurypharynx pelicanoides, called also the pelican eel. To describe the jaw and have you understand what you are actually hearing requires setting aside every reference your experience with fish has given you. The mouth of the gulper eel can open wider than the total length of the animal itself, not the width of the head, not the width of the body, the total nose to tail length of the fish. The lower jaw is not hinged in any way that resembles surface fish anatomy. It swings downward, then outward, then further outward still. The gape it creates is not simply a large mouth. It is a different category of opening entirely. The interior is a void capable of taking in prey considerably larger than the predator's own head. And the stomach behind the jaw was built to receive whatever the jaw delivers. When a gulper eel feeds, the body changes shape visibly and dramatically. The translucent stomach distends outward in all directions. The creature that was a narrow, elongated form with a large mouth in front becomes something with a swelling mass behind it, a meal visible from outside the body. In the complete darkness where this happens, it goes unseen by everything except the next predator, unfortunate enough to encounter a recently fed gulper moving slowly through the water. The hunting behavior matches the anatomy. The gulper eel does not pursue, it does not sprint. It opens its mouth and moves forward at the slowest metabolically viable speed through the dark water. It is a living net. Moving slowly burns fewer calories. Burning fewer calories means the last meal supports the body longer. The mouth simply needs to be open and present when something edible passes through the water column the eel is occupying. The patience and the mouth together make a system that is devastatingly effective for an animal that cannot afford to chase anything. At the tip of the gulper eel's tail is a small bioluminescent organ producing a pink-red glow. Whether this lures curious prey toward the open mouth at the other end or serves another signaling function is still investigated. What is clear is that the gulper eel invested bioluminescence at both ends of its body. The tail attracts, the mouth receives, the stomach accommodates. Now meet the black swallower. Chiasmodon niger is small. Adults measure between 7 and 25 centimeters. Nothing in its external appearance signals what it is capable of. No spectacular structural modification visible from outside. No fangs extending past the body. No transparent dome or glowing lures. Just a small dark fish. The extreme feature is entirely interior. The stomach. The black swallower's stomach can extend to hold prey more than twice the animal's own total length, 10 times its own body mass. The stomach walls are not rigid tissue. They are elastic in all directions simultaneously. The fish's skin itself stretches to accommodate what is inside. Internal organs compress and shift to make room. The animal expands around its meal and temporarily exists in a different size class than the one it biologically belongs to. We know this because of how black swallowers were first documented in significant numbers. They were found floating at the ocean's surface, dead, transparent, distended stomachs visible from outside the body. Inside those stomachs were prey animals in various states of decomposition. The sequence of events that produced this picture is straightforward once you understand the mechanism. The swallower had taken in a meal too large to digest quickly. The prey began decomposing chemically inside the swallower before digestion could complete it. Chemical decomposition produced gases. The gases inflated the elastic stomach. The inflated stomach created buoyancy. The dying swallower rose through thousands of meters of dark water and arrived at the surface still containing its last meal in an enormous gas-filled transparent pouch. We found them drifting up from the dark, killed by the ambition of a single meal. The loose-jawed dragonfish rounds out this chapter with a different approach entirely. Not maximum capacity, but invisibility of the strike itself. The lower jaw of Malacosteus niger has no membrane spanning its floor. The structure that in almost every other fish seals the base of the lower jaw simply does not exist. The jaw is bare bone. When this jaw swings open below prey in complete darkness, there is almost nothing for the prey to detect approaching from below. No solid surface, no closing shadow, no movement of tissue that triggers the prey's lateral line sensing system. By the time the jaw completes its motion around the prey, the encounter is already decided. There is a fourth creature in this zone that deserves a moment of its own. The stoplight, loose-jaw Aristostomius scintillans, takes the concept of the invisible strike and combines it with a predatory posture so unusual it looks like a design error until you understand the logic. The lower jaw hangs open at rest, not partially, fully. The jaw is positioned open by default because closing it takes muscle energy the fish cannot spare between meals. The stoplight loose jaw moves through the water with its mouth already open, ready, patient in a way that goes beyond behavior into physiology. The body conserves every calorie by not performing even the small muscular act of keeping the jaw closed when there is nothing to close it around. Think about what that means. An animal so adapted to the arithmetic of deep-sea starvation that it evolved a resting mouth position of open rather than closed. Because open is the productive state and closed is the state of waiting. And in the deep ocean, the less time spent waiting, the better. These creatures together illustrate something that cuts across all the biology in this chapter. The deep ocean did not produce extreme mouths because evolution was excessive. It produced them because the food situation at depth is genuinely extreme enough to justify every one of them. The gulper mouth, the elastic stomach, the invisible jaw, the permanently open resting posture. Each one of these features is the precise response to the same underlying equation. Food is rare and unpredictable, and every encounter with it may be the last for weeks. The body that fails to take advantage of each one will eventually pay for that failure. The body that succeeds passes on its feeding architecture to the next generation. Over millions of generations of that selection, the deep ocean produces mouths that look like nightmares, because nightmares are what the food supply requires. The deep ocean built these mouths for one reason. In the absence of abundance, every body must become a perfect trap. Every meal that escapes is a deficit the predator may not recover from. In the next chapter, we go deeper still, where the sun has never reached. We meet a creature that built the most elaborate trap in the deep ocean, one that runs on light and patience, and the universal attraction every living thing feels towards something glowing in the dark. Chapter 5. The Lure of Death, How the Anglerfish Hunts. There is a moment in the descent into the deep ocean where the last trace of sunlight disappears completely. Not fades, not dims to imperceptibility, disappears. At approximately 1,000 meters, the final photon from the sun ceases to exist in the water column. Below this depth, no instrument has ever recorded a single particle of light arriving from the surface above. The midnight zone begins here, the bathypelagic region between 1,000 and 4,000 meters, where pressure climbs from 100 to 400 atmospheres, where the temperature stabilizes just above freezing in most regions, where the water is cold enough that chemical reactions slow, metabolism slow, everything slows. The entire environment runs at a reduced pace, dictated by conditions that allow very little speed and very little warmth. In this environment, evolution had to solve a problem that became almost philosophically extreme. How do you find food in a world where you cannot see, where your prey cannot see, and where the distances between anything edible stretch into hundreds of meters of empty dark water in every direction? One family of fish found an answer so counterintuitive that when scientists first encountered it, they could barely accept an organism had arrived at it independently. Instead of hiding from prey, instead of becoming invisible or patient or still, the anglerfish became visible deliberately, conspicuously. It manufactured a light in the darkness and waited for the darkness to deliver everything toward it. The anglerfish, the serratioid anglerfish, a family of approximately 200 species distributed across the midnight zone. When most people picture an anglerfish, they picture the female. They should. The female is the hunter. The female is the creature that evolved the esker. The female is the one that built the most sophisticated predatory architecture in the midnight zone. The female serratioid anglerfish is not large by any standard, except the context of where it lives. Most species reach between 20 and 30 centimeters. The body is roughly spherical, wide, soft, dark. The skin texture is smooth or lightly patterned, and its color is typically dark brown or black, because in the midnight zone, no pigmentation serves any visual camouflage purpose. The mouth occupies a disproportionate percentage of the head. When closed, it is already a striking feature. When open, it reveals something that belongs in a different chapter of this documentary entirely. The jaws are lined with teeth, long, transparent teeth that curve inward toward the back of the mouth rather than outward. These are not teeth designed to sever. They are not designed to cut prey into pieces the way a predator on the surface world might process a meal. They are designed to prevent exit. Once something passes between those teeth, it encounters a geometry that becomes more confining with every millimeter of forward movement. The space between the teeth is widest at the entrance. It narrows as it deepens. They are a cage with a closing door. Something can enter. Nothing leaves under its own power. But the teeth are not what delivers prey to the cage. The esker is. The esker is a modified dorsal fin ray. On surface fish, a dorsal fin ray is a structural element of the back fin. In the female serratioid anglerfish, one of those rays migrated over millions of years of evolution forward along the body, toward the head, and then beyond the head, until it extended from the front of the snout like a fishing line hanging from a pole built into the skull. At the very tip of that line is a small bulbous organ, and inside that organ lives light. The esker glows. In the absolute darkness of the midnight zone, it produces a blue-green bioluminescence that is the only visible light in the immediate environment for some significant distance in any direction. It pulses with a rhythm the anglerfish controls. through fine muscular adjustments to the lure's supporting structure. The esker can swing gently, it can pulse rapidly or slowly. It can drift with the movement of a small organism floating passively in the water column. It is a performance, deliberate and calibrated, and aimed at one specific and continuously changing audience. But the anglerfish does not produce this light herself. She cannot. No serratioid anglerfish produces its own bioluminescence directly. The esker houses a colony of symbiotic bioluminescent bacteria from genera related to Photobacterium. These bacteria produce the light biochemically using the enzyme luciferase in a reaction that generates photons without generating heat. The anglerfish provides the bacteria with shelter inside the esker tissue and with nutrients delivered through the bloodstream. The bacteria provide the glow that powers the entire hunting system. The relationship is one of total mutual dependency. Without the bacteria, the esker goes dark and the hunting system fails. Without the anglerfish, the bacteria have no host. The light in the darkness of the midnight zone is not her creation. It is a borrowed fire she maintains inside her own body. Why does the light work so well? In the midnight zone, the relationship between light and life is different from anywhere else on the planet. Below 1,000 meters, every creature that exists in the water column has evolved over millions of years in the presence of only two categories of light: the bioluminescent flashes of other organisms and complete darkness. There is no ambient glow to provide context. There is no gradient of illumination that tells a creature where safe hiding places are. There is only the dark and occasionally something glowing in it. When a deep-sea organism detects a glowing point of light in that environment, it faces a calculation with very few variables. The light is biological. Biological things in the midnight zone are either food or predators. Food is rare and metabolically essential. The olfactory and visual systems of most midnight zone creatures are calibrated to respond to bioluminescent signals. The esker exploits exactly this calibration. It does not just glow, it glows in the precise spectrum and with the precise pulsing behavior that triggers the approach response in the prey species that share its environment. The trap is built from the prey animal's own instincts. The hunting strategy that follows from this arrangement is one of the most patient in the deep ocean. The female positions herself in the dark water and holds still. She moves as little as possible. Movement costs energy, and energy at these depths comes from meals that may be weeks or months apart. She holds position. She pulses the esker, small fish, shrimp, copepods, anything in the surrounding darkness that registers the light and turns toward it. Because in the midnight zone, moving toward a light source is an instinct that evolution has not suppressed in most species. Light means something biological. Something biological at these depths might mean food. The prey approaches. The moment the prey is within range, the anglerfish strikes. The jaws snap closed at a speed researchers describe as among the fastest jaw movements of any known fish. The transparent inward curving teeth close around whatever is inside them. The cage is shut. The anglerfish then does something that requires its own description. The prey is held by the teeth. The swallowing process begins. The jaw structure of the serratioid anglerfish has evolved to allow objects larger than the resting mouth opening to be ingested by a sequential process of jaw articulation that gradually moves the prey backward. The head of the prey enters first. The teeth release one side at a time and re-engage further back. The prey moves through the throat in stages. The process can take minutes or longer depending on size. The anglerfish remains essentially motionless throughout. All available energy is directed toward the intake of the meal. Nothing is wasted on movement. And if the prey exceeds the size of the predator's resting body, the stomach stretches. The elastic tissue of the stomach expands in all directions to accommodate prey that the anglerfish's external appearance would suggest she could not possibly contain. Scientists have documented female serratioid anglerfish with prey inside them that should have been physically impossible to pass through the head. Prey that was swallowed whole and is visible as a solid mass, distending the body outward in all directions. In the midnight zone, prey is too rare to decline for any reason, including size. She is the trap. The eska is the invitation. The darkness is the theater. In the next chapter, we meet the male of this species and discover that for the serratioid anglerfish, survival takes a form that has no parallel anywhere else in the vertebrate world. Chapter 6. Flesh Made One. Sexual Parasitism and the Body Horror of Deep-Sea Reproduction. In the previous chapter, we met the female serratioid anglerfish, the hunter, the creature with the eska, the one with the elastic stomach and the transparent cage teeth and the borrowed bacterial light. We described her as the most efficient predatory system in the midnight zone. That description is accurate, but it is incomplete, because there is a male of this species, and the male tells a completely different story, a story that has no parallel anywhere else in the vertebrate world, a story that crosses boundaries between separate organisms that the science of biology had not previously considered crossable. A story that begins with one of the most extreme mismatches in size between male and female in any vertebrate species on this planet, and ends with something that should not be biologically possible. Let us start with what the male serratioid anglerfish is. He is small, dramatically small. Depending on the species, the male is anywhere from 6 to 40 times smaller than the female. The female we described in the previous chapter, the 20 to 30 centimeter creature, is a giant compared to the male. A mature male serratioid anglerfish in most species measures between 6 and 10 millimeters. Some species produce males barely 3 millimeters in total length. He is not a smaller version of the female. He is a fundamentally different organism, built for a fundamentally different purpose. The female has a large functional jaw for feeding. The male has a reduced jaw structure not designed for serious predation. The female has a fully developed digestive system that can accommodate prey twice her own size. The male has a simplified digestive system barely capable of sustaining him through his one assignment. The female has the eska with its bacterial light colony and its fine muscular controls. The male has enormous olfactory organs, occupying a significant proportion of his head. Organs of smell so sensitive and so developed relative to his body size that they represent one of the most extreme chemosensory adaptations in any vertebrate. The male cannot hunt the way the female hunts. The male was not built to hunt. The male was built to follow a scent. The female releases specific chemical compounds into the water, pheromones. The male has evolved to detect these compounds at extraordinarily low concentrations across the vast and lightless distances of the midnight zone. When he detects the signal, he turns toward it and he swims. He swims and he follows the chemical trail through the darkness. He has almost no other purpose during the brief independent phase of his life. He does not need to eat much. He does not need to see well. He does not need complex behaviors. He needs one thing: to reach the source of the scent. When he finds the female, he bites her. Not an aggressive bite, not a feeding bite. He bites into the skin of her body and he holds on. This is the beginning of something that has no equivalent in vertebrate biology before or since. The male releases enzymes from tissue around his mouth as he holds on. These enzymes begin to dissolve the cellular boundary between his tissue and hers. They break down the membrane between his skin and the skin of the female at the site of contact. The cells at the junction begin to merge. Blood vessels on both sides grow toward each other. Through the dissolving boundary, through the chemical pathway the enzymes created, the blood vessels of the male and the female find each other and connect. The male becomes physically connected to the female at the circulatory level. His blood and her blood now flow through a shared system. The nutrients she absorbs from her meals begin to feed him through the merged bloodstream. He no longer needs to find food himself. He no longer needs to swim in search of anything. He is attached. He is fed. He has arrived at the only destination his biology was constructed to reach. And then his body begins to change. With the connection to the female established and nutrition flowing from her blood to his, the male's own organs lose their purpose. Evolution operates on a principle that unused structures are expensive and should be reduced. The male's eyes, already small and not highly functional, begin to degenerate over the weeks and months following fusion. The lenses shrink. The visual processing capability declines. Eventually, the eyes are vestigial structures with no meaningful function remaining. The male is now blind. He does not need to see. He is attached. The internal organs follow. The digestive system that sustained him during his brief independent phase degenerates. The circulatory organs simplify. The musculature, beyond what is needed to maintain attachment, reduces. The male becomes, over time, biologically simpler. Organ by organ, his body returns the energy investment of maintaining complex tissue to the shared system. He is being deconstructed in place. What remains? The testes. The testes are the one structure that does not degenerate. They are the reason the male existed. They are the reason he followed the scent. They are the reason the enzymes were released and the blood vessels merged and the organs were deconstructed. The testes remain functional. They produce sperm. The female carries them. When the female is ready to reproduce, the sperm are available. The male, whose eyes and gut and muscles have long since dissolved into the shared tissue, provides the one thing he was built to provide. A female serratioid anglerfish can carry multiple males simultaneously. Some specimens have been found with 6 or more males fused to different parts of the body. Each one arrived at a different time. Each one followed his own chemical trail through the darkness to the same source. Each one bit. Each one fused. Each one is now part of her body in a way that blurs the boundary between two separate organisms almost completely. This is the only known example of genuine physiological fusion between two separate vertebrate organisms in the history of life on this planet. Not a relationship. Not a symbiosis maintained by proximity. An actual merger of tissue and blood and ongoing biological function across what was once a boundary between two distinct animals. There is a scientific detail about the fusion that deserves its own moment because it changes how you think about what biology is capable of. When the male bites the female and the enzymes begin dissolving the tissue boundary, the immune systems of both organisms face a crisis. The immune system exists to identify and attack foreign tissue, foreign cells. Anything that does not carry the organism's own molecular identity markers. In virtually every other vertebrate organism on Earth, when tissue from one individual contacts tissue from another individual, the immune system responds by attacking and destroying the foreign material. Organ transplants fail without immunosuppressant drugs for exactly this reason. Two organisms of the same species still have immune systems that recognize each other as foreign. In the serratioid anglerfish, both immune systems suppress the rejection response at the site of fusion. The mechanism by which this happens is not yet fully understood and is one of the most actively studied aspects of their biology. What is known is that neither fish destroys the other's tissue. Neither immune system attacks. The fusion proceeds. The blood vessels merge. The bodies integrate. Two organisms become one compound organism that contains the functional biology of both. Immunologists who study the serratioid anglerfish regard this as one of the most remarkable examples of immune tolerance in vertebrate biology. It is not clear whether the suppression mechanism evolved in the male, the female, or both simultaneously. What is clear is that it works, and that without it the entire reproductive strategy of this species would fail at the moment of contact. Why did it evolve? The midnight zone provides the answer. Food is rare. Encounters between individuals of the same species are rarer still. In the vast and dark water column between 1,000 and 4,000 meters, two anglerfish may pass within range of each other, once, only once, in an entire lifetime. The cost of missing that meeting, of failing to reproduce on the one occasion the opportunity presents itself, is absolute. There is no next time. There is no second chance at depth. The male who fused passed on his genes. The male who did not find a female did not. The merger was not an accident of biology. It was the most rational possible response to an environment where every resource, including the proximity of another member of your own species, is more precious than it is anywhere else on this planet. In the next chapter, we leave the anglerfish and descend further, to meet the creatures that solved the problem of being seen in a completely different way, not by attracting prey with light, but by becoming something that light itself cannot find. Chapter 7. The Invisible Killers, Ultra-Black Fish and Transparent Predators. In the midnight zone, two opposite strategies evolved to solve the same problem. The problem is visibility. In an environment where 75 to 80% of all organisms produce bioluminescent light, the challenge of not being seen is more complex than it would be in a simply dark environment. Darkness alone would make camouflage easy, but the deep ocean is not simply dark. It is dark with intermittent cold fire moving through it in every direction. Flashes, pulses, searchlights, lures. The bioluminescent output of thousands of organisms in any given volume of midnight zone water means that any creature moving through that volume is constantly at risk of being briefly illuminated by the light of something nearby. For a predator, this creates a specific problem. If a predator searching for prey illuminates the water column with its own bioluminescence, a perfectly black target reflects less of that light back than any other surface. And if the target is transparent, it reflects almost nothing at all. Both approaches produce invisibility, but they produce it through completely different physical mechanisms. And they were evolved independently by dozens of separate lineages across the deep ocean. In 2020, a team of researchers from Duke University and the Smithsonian Institution published a study in the journal Current Biology that changed how the scientific community understood darkness in the ocean. The researchers, led by Alexander Davis and Karen Osborne, were studying specimens of deep-sea fish when they noticed something unusual about certain species. Their skin appeared to absorb light in a way that exceeded what standard biological pigmentation should be capable of producing. They measured it. 16 species of deep-sea fish absorbed more than 99.5% of all light that struck them. Standard black paint, the blackest material most humans encounter, reflects approximately 2% of incident light. The blackest human-made materials at the time of the study reflected between 0.5% and 1%. These 16 species of fish reflected less than 0.5%. Some reflected as little than 0.05%. They were absorbing light at a rate 20 times more effective than the darkest pigmented materials humans had manufactured at that point. The Pacific black dragon, Idiocanthus antrostomus, was among the species confirmed. The fangtooth Anaplogaster cornuta appeared again. An animal that had already featured in this documentary for its extraordinary teeth was now revealed to also carry one of the most extreme light-absorbing surfaces in the biological world. The same creature that evolved skull sockets to receive its own fangs also evolved skin that is effectively a black hole for visible light. How does the skin achieve this? The answer is in the melanosomes. Melanosomes are the organelles that produce and contain melanin, the pigment responsible for dark coloration in skin, hair, and eyes across the animal kingdom. In most pigmented animals, melanosomes are relatively small and are scattered through multiple tissue layers. They absorb light that reaches them but allow some light to pass between them and some to scatter in ways that eventually exit the tissue surface. In ultra-black deep-sea fish, the melanosomes are different. They are larger, more disc-shaped than the melanosomes found in surface animals. They are packed into a single dense layer immediately beneath the surface of the skin. And because of their specific geometry, they do something unusual when light strikes them. Instead of absorbing what they can and letting the rest pass or scatter outward, the arrangement causes photons to scatter laterally within the melanosome layer. A photon that hits one melanosome and is not absorbed scatters sideways into an adjacent melanosome. And if that one does not absorb it either, it scatters again into the next. The photon essentially bounces around inside the layer of melanosomes. Each bounce is another absorption opportunity. The system does not give light a path back out. It is an optical trap. Light enters. Light does not exit. The fish effectively built an absorbing surface from its own biological material using a packing geometry that has since inspired research into human-engineered ultra-black materials. The deep sea invented this manufacturing principle independently and significantly earlier. The researchers studied the ultra-black specimens using electron microscopy to examine the melanosome layer at the nanoscale. What they found was that the packing density and orientation of the melanosomes appeared to be optimized in a way that maximized the lateral scattering behavior. It was not simply that there were more melanosomes per unit area than in other fish. It was that their specific shape and arrangement created an architecture with optical properties more extreme than any other biological pigmentation system previously documented. The researchers estimated that the geometry of the melanosome layer in these fish approaches the theoretical maximum absorption efficiency achievable through biological melanin. What drives the evolution of this extreme darkness? The answer connects back to the same bioluminescent arms race that shaped eyes and jaws and lures throughout this documentary. The deep-sea predators that hunt using bioluminescent searchlights are searching for reflected signals. A fish that reflects 2% of the light striking, it is 200 times more visible than a fish that reflects 0.01%. In an environment where the difference between being detected and not being detected determines survival, the evolutionary pressure toward lower and lower reflectance is relentless. The ultra-black fish did not evolve their skin color for the same reasons surface animals evolved pigmentation. They evolved it to disappear from the hunting beams of other creatures. Their darkness is not aesthetic, it is armor. But not every deep-sea creature solved the problem of visibility by absorbing light. Some solved it by refusing to interact with light at all. The glass octopus, Vitreledonella richardi, is an octopus found in the tropical and subtropical open ocean at depths ranging from several hundred to over 1,000 meters. Its body is almost completely transparent. The mantle, the arms, the internal organs including the digestive gland, the musculature, all are transparent or nearly so. The only visible structures are the eyes and the optic nerves and a small section of the digestive tract. Everything else passes light through without meaningful reflection or absorption. A predator illuminating the water column and searching for the reflected signature of prey finds almost nothing when that light passes through a glass octopus. The cystosoma amphipod takes this strategy even further. This crustacean, found in deep mesopelagic and bathypelagic waters, is one of the most completely transparent animals in the ocean. Its body appears as little more than a faint geometric outline in any light directed at it. The internal organs are enclosed in tissue that scatters almost no light. Even the eyes in some specimens are reduced or modified to minimize the reflective signature they create. Why does transparency work as a camouflage strategy when it seems like the opposite of hiding? The answer is that camouflage in the deep ocean is not about matching a background color. There is no background color to match. The challenge is to produce no optical signal that a predator or prey can detect. A perfectly transparent object produces almost no reflected light signal and no shadow in diffuse illumination. In the dark water column, where light arrives as intermittent bioluminescent pulses rather than steady ambient illumination, a transparent body becomes essentially a ghost. It is there in the water, but nothing about it triggers the visual systems of the organisms sharing its environment. Two strategies, ultra-black and transparent, born from the same environment and the same pressure. One absorbs all light, one transmits all light. Both arrive at the same destination, invisible. The existence of both strategies in the same environment tells us something important about the nature of evolutionary solutions. There is rarely one right answer to a biological problem. The deep ocean presented its inhabitants with the challenge of invisibility, and the deep ocean produced not one solution but two fundamentally different engineering approaches that each work completely. The ultra-black fish took the light and swallowed it. The transparent creatures stepped aside and let it pass through. Both are invisible to the same predators for entirely different physical reasons. In the next chapter, we stop descending for a moment, and instead of going deeper, we look at what time does to the deep ocean. We meet the creatures that evolution forgot to change, the animals that should not still exist, the living fossils of the deep. Chapter 8. Things that should not exist. Goblin sharks, frilled sharks, and living fossils. The deep ocean is not only a place where evolution pushes biology forward toward new extremes, it is also a place where evolution stands still. On the surface world, the conditions of life change constantly. Climates shift, sea levels rise and fall, ice ages advance and retreat. Forest becomes desert, becomes ocean. The surface is dynamic. And in a dynamic environment, species that cannot adapt disappear. The fossil record of the surface world is a long record of extinction and replacement, of lineages that ran out of time to adjust. The deep ocean is different. At 4,000 meters, the temperature has been stable near freezing for tens of millions of years. The pressure has not changed. The darkness has not changed. The sparse food supply has not changed. For some lineages living in the deep ocean, the environment they occupy today is essentially the same environment their ancestors occupied 200 million years ago. In a world that never changes, there is no pressure to change. And so, some of the creatures that evolved for the deep ocean look today almost exactly as they looked when the dinosaurs were the dominant land animals. They are not primitive. They are not failures of evolution. They are organisms so well adapted to conditions that never changed that adaptation became unnecessary. They are the deep ocean's most extraordinary ambassadors to the present day, and they look like things that should not still exist. The goblin shark, Mitsukurina austoni, is one of the rarest large animals on Earth. Fewer than 50 complete specimens have ever been collected by science. It lives at depths ranging from approximately 200 to over 1,000 meters across the Atlantic, Pacific, and Indian Oceans. Its lineage traces back at least 125 million years in the fossil record. The oldest known ancestor of the goblin shark swam in the oceans of the Cretaceous period. In a time of ammonites and mosasaurs and an ocean that no longer exists in its former geography, the goblin shark looks precisely as ancient as it is. The snout extends forward from the head in a long, flat blade covered in sensory organs called ampullae of Lorenzini. These organs detect the electrical fields produced by the muscular contractions of living animals in the surrounding water. The goblin shark uses this electrical sense to locate prey in the dark where vision is limited. The snout is not used for attack. It is a sensory array, a biological instrument panel extended in front of a head built to process its output. The jaw is what makes the goblin shark memorable. At rest, the jaw is retracted beneath the snout, giving the face an unusual recessed expression. But when the goblin shark detects prey, it releases the jaw from its resting position. What follows is called slingshot feeding. The jaw does not simply open, it fires forward. The entire jaw structure projects outward from beneath the snout at speed, driven by elastic tissue that releases stored energy in the same way a catapult releases stored tension. The jaw extends to a point that brings the teeth almost level with the tip of the long snout. The strike is completed. The jaw retracts. The whole sequence takes approximately 300 milliseconds. The goblin shark's skin appears pink or reddish because the tissue is thin enough to show the blood vessels beneath. In the dark of its environment, coloration is irrelevant. The skin did not evolve for appearance. It evolved for function alone. The result is an animal that looks to us like something between a creature of the deep and a creature from before the deep was the deep. A face assembled from geological time rather than ordinary evolutionary time. Now meet the frilled shark. Chlamydoselacus anguineus was first described to Western science in the 1880s. Its lineage is even older than the goblin shark. Frilled shark ancestors appear in the fossil record from the Cretaceous, and some researchers argue the broader group extends back into even older geological time. The frilled shark today is so anatomically similar to its fossil ancestors that it is sometimes called a living fossil. That term is debated in biology because it implies a creature stopped evolving, which is never quite accurate. But the gap between the frilled shark's appearance and the appearance of animals known only from ancient rock is genuinely small. The body of the frilled shark is elongated, eel-like. It reaches lengths of approximately 2 meters. The head sits at the front of this long body with a mouth that opens at the very front rather than underneath as in most sharks. The teeth are extraordinary. The frilled shark carries approximately 300 teeth arranged in 25 rows. Each tooth has 3 points separated by small cusps. They do not look like the simple triangular blades most people associate with shark teeth. They look like something that belongs in a different kingdom of life entirely. The frilled shark hunts by coiling its body and striking forward in a motion that researchers compare to a snake striking. It targets soft-bodied prey, including squid and small fish. Its long, flexible body allows it to maneuver in ways that rigid-bodied sharks cannot. It is a design that worked well enough in the Cretaceous that the ocean of today still contains it. Living specimens of the frilled shark are rarely encountered. The animal was long known only from occasional specimens washed ashore or caught accidentally in deep fishing nets. A filmed specimen swimming in shallow water off Japan in 2007 briefly appeared in news coverage around the world before dying. The footage showed an animal that moved with a fluidity unlike any modern shark, something that looked borrowed from a different era of the ocean. There is a third creature from this category of deep ocean living relics that deserves mention alongside the goblin shark and the frilled shark. Not because it is equally dramatic in appearance, but because its rediscovery in 1938 was one of the most significant moments in 20th century natural history. The coelacanth, Latimeria chalumnae, was known only from fossils before 1938. The fossil record extended back approximately 400 million years and then stopped. Scientists assumed the coelacanth had been extinct for 65 million years, part of the Great Cretaceous extinction event that ended the dinosaurs. Then, a museum curator in South Africa noticed an unusual fish among the daily catch at a local fish market. A fish with fleshy lobed fins that looked like proto-limbs. A fish that matched the fossil record precisely. The coelacanth had not been extinct. It had been living at depths of 100 and 700 meters along the coasts of the Comoros Islands, in the Indian Ocean and in Indonesian waters. An animal from the age of early life on Earth had simply retreated to deep water and continued existing without interruption. It had been in the ocean all along. We had simply not been looking in the right place. The coelacanth is not the only example. It is simply the most famous. The deep ocean contains dozens of lineages with similarly deep roots in geological time. Creatures that the fossil record suggested were gone, that continued living at depth in conditions too stable to demand change. The lesson the deep ocean teaches through these animals is a humbling one. We do not know what is down there. Our surveys of the deep have covered a fraction of a percent of its total volume. The ocean is enormous and our instruments are new. The living fossils we have found are the ones that occasionally come close enough to the surface or to fishing operations to be accidentally captured. How many more ancient lineages persist in the dark at depths we have never surveyed with adequate equipment is a question the ocean has not yet answered. What both of these creatures demonstrate is that the deep ocean functions as a biological archive. A cold, dark, stable repository where lineages that would have been eliminated by surface conditions found protection in unchanging depth. The goblin shark and the frilled shark are not simply unusual animals. They are time travelers, organisms that arrived in the present day from geological eras so distant that the surface world they might have shared with their ancestors is unrecognizable. They survived not by adapting to a changing world, but by choosing a world that did not change. In the next chapter, we meet the creatures at the opposite end of this spectrum. Not the survivors from the ancient past, but the organisms that the deep ocean drove to become the largest of their kind that has ever existed. Chapter 9. The Giants, Deep-Sea Gigantism, and the Creatures that Grew Too Large. Something strange happens to size in the deep ocean. Crustaceans that would fit in your hand on a coral reef grow to the size of footballs in the deep. Worms that measure centimeters on a beach surface grow to meters in the abyss. Squid that are modest in shallow water grow to lengths that exceed the largest known fish. The pattern appears across dozens of unrelated lineages and has been observed consistently enough that biologists gave it a name. Deep-sea gigantism. The tendency for organisms in deep or cold water to grow substantially larger than their closest relatives in shallower, warmer environments. The causes of deep-sea gigantism are still debated. Multiple factors appear to contribute: cold temperatures, slow metabolism, and may allow individuals to live longer without the wear of heat-driven biochemistry. Longer lives mean more growth time. The extreme scarcity of food may favor larger body size because larger bodies can store more energy between meals. Lower levels of dissolved oxygen at certain depths may affect the hormonal systems that regulate body size. Whatever the mechanism, the effect is real, and it produces some of the most extreme examples of biological scaling on this planet. No creature illustrates this more dramatically than the giant squid. Architeuthis, ducks, the giant squid. Adults reach documented total lengths of up to 13 meters, including the 2 long tentacles that extend beyond the 8 main arms. The mantle alone, the main body structure, can reach 2 meters in length in the largest confirmed specimens. The weight of a fully grown adult is estimated between 100 and 250 kilograms. The giant squid is the largest invertebrate on Earth. But the feature of the giant squid that science regards as its most extraordinary is not its length or its weight. It is the eyes. The eyes of the giant squid reach up to 27 centimeters in diameter. 27 centimeters. Roughly the diameter of a dinner plate. They are the largest eyes of any living animal on this planet. They are large for the same reason all deep-sea eyes tend towards size: to capture every available photon of light in an environment where light is either absent or produced only in brief bioluminescent bursts. But the giant squid takes this to a scale that has no equivalent in any other living organism. Eyes that evolved to see in the dark and ended up becoming the largest light-gathering structures in the animal kingdom. For most of the 20th century, the giant squid was known almost entirely from dead specimens. Tentacles found in the stomachs of sperm whales, partial carcasses that drifted to the surface or washed ashore, circular sucker scars on sperm whale skin, suggesting battles between the 2 species at depth. The giant squid was known to exist, but had never been photographed alive in its natural habitat. It was a real creature that felt mythological because the only evidence of it was fragmentary and always post-mortem. In 2004, a research team led by Tsunemi Kubodera and Kyoichi Mori deployed a camera baited with a smaller squid at depth in the North Pacific. They obtained the first photographs of a living giant squid in the wild. The images showed an animal both recognizable from preserved specimens and deeply strange in motion, active, alert. Not the passive drifting creature some had imagined, but a fast and deliberate predator. The footage that followed in subsequent years confirmed that the giant squid moves with a fluency in the deep ocean that its preserved remains had not suggested. Its relative, the colossal squid, is heavier still. Mesonychoteotis hamiltoni, the colossal squid. Adults are estimated to exceed 500 kilograms in mass, making them the largest invertebrates by weight ever recorded. The tentacles are shorter than those of the giant squid, but the body mass is substantially greater. The colossal squid carries something on its tentacles that the giant squid does not: rotating hooks. Sharp, rigid structures embedded in the suckers of its tentacles that spin and can be driven into prey tissue when the tentacle coils around a target. It is a different predatory architecture built into the same general body plan. The colossal squid is so rarely encountered that our understanding of it comes almost entirely from specimens recovered from the stomachs of sperm whales. The deepest hunting marine mammals on the planet are apparently hunting these animals regularly, which tells us that in the deep southern ocean around Antarctica, colossal squid are present in numbers large enough to support sustained sperm whale predation. We simply cannot get cameras to where they live in sufficient numbers to see it directly. The giant isopod brings deep-sea gigantism to crustaceans. Bathynomus giganteus is a crustacean related to the small pillbugs and woodlice found in gardens around the world. Those surface relatives are typically 1 to 3 centimeters long. The giant isopod reaches up to 50 centimeters in length and can weigh over 1.5 kilograms. It is the largest known crustacean of its order. It lives at depths between 170 and 2,500 meters on the seafloor, where it scavenges dead organisms, including the rare large food falls like whale carcasses. The giant isopod can fast for extraordinary periods. Specimens in aquariums have survived without food for over 4 years. The metabolism runs at such a slow rate, and the energy storage in the body is sufficient enough that starvation that would kill a surface animal within weeks is simply a resting state for the giant isopod. It waits. For weeks, for months, for years if necessary. And when food arrives in the form of a dead whale or large fish on the seafloor, it feeds aggressively and rapidly before settling back into its patient economy. The giant spider crab of the deep temperate Pacific, Macrochera caempheri, extends the gigantism story into arthropods with legs. A fully extended adult can span 3.7 meters from the tip of one front claw to the tip of the other. The body is relatively compact. The legs are extraordinarily long and thin. In motion across the seafloor, the giant spider crab moves with a slow, deliberate precision that is more reminiscent of a robot navigating rough terrain than of anything we usually associate with a crab. It is the largest arthropod alive, larger than any lobster, larger than any horseshoe crab, larger than any surface-dwelling relative by a factor that reflects millions of years of cold water growth without the constraints that warm environments impose on body size. There is also the giant oarfish, Regalecus glesner, the longest bony fish in the world. It lives at depths ranging from 200 to 1,000 meters and reaches documented lengths of up to 8 meters with unconfirmed reports of specimens exceeding 11 meters. The oarfish has a long silver body with a distinctive red dorsal fin that runs its entire length. It moves through the water in undulating vertical motions completely unlike any surface fish. For centuries, dead oarfish washing ashore were the source of sea serpent legends from Norway to Japan. The creature that inspired the mythology of enormous serpentine sea monsters was not imaginary. It was simply not being observed at depth where it actually lives. The pattern of gigantism in the deep ocean is not confined to these headline examples. Amphipods that measure a few millimeters in shallow water reach 25 centimeters in the deep ocean. Sea spiders the size of dinner plates patrol the abyssal floor. Tube worms in vent communities reach lengths of 2 meters. In every taxonomic group that spans both shallow and deep environments, the deep representatives are larger, often dramatically so. The cold and the dark and the strange economies of the deep ocean do not simply change what life looks like. They change what size means. These three creatures, the giant squid, the colossal squid, and the giant isopod, are the most visible examples of a pattern that runs through dozens of deep-sea lineages. The deep ocean takes the body plans of surface animals and scales them toward extremes. It does this through cold and time and the particular demands of surviving in an environment where size is an advantage that compounds over the very long lifespans that the deep ocean makes possible. In the next chapter, we meet one of the most ancient and most unusual creatures in the midnight zone. An animal that is not quite a squid and not quite an octopus, and that has been living in the oxygen-depleted darkness since before most of the ocean was the ocean it is today. Chapter 10. The Vampire of the Void. Ancient Lineages and Creatures from Before Memory. In the ocean there is a zone that most life avoids. Not the deep because of pressure, or the midnight zone because of darkness. A zone that is inhospitable because of something more subtle and more chemically fundamental, the oxygen minimum zone. A layer of ocean water typically found between 200 and 1,000 meters depth in certain regions of the Pacific and Indian Oceans, where the concentration of dissolved oxygen drops to levels that are lethal for most aerobic life. The oxygen minimum zone forms because organic matter sinking from the surface above is decomposed by bacteria that consume oxygen faster than it can be replenished by ocean circulation. The result is a band of near anoxic water that most fish and crustaceans and squid cannot tolerate. One creature evolved to make the oxygen minimum zone its home. Vampiroteuthis infernalis, the vampire squid from hell. That is not a dramatic nickname, that is its scientific name. Vampiroteuthis, vampire squid, infernalis, from hell. The scientists who named it in 1903 were apparently not given to understatement. The vampire squid is not a true squid. It is not a true octopus. It belongs to its own separate order, Vampiromorphida, an order that contains only one living species, Vampiroteuthis infernalis. Its closest known relatives are known only from fossils. The order diverged from the common ancestor of squid and octopus deep in geological time, and the lineage that produced this single living species has been following its own evolutionary path ever since. The vampire squid is genuinely alone in its taxonomic category, the last surviving member of an ancient branch of cephalopod evolution that all other lineages left behind. It reaches a maximum length of about 30 centimeters from the tip of the mantle to the end of the longest arms. It is not large, but its appearance in any light is striking. The body is deep red to black in color. The webbing between the arms forms a cloak that the animal can pull up over itself when threatened. Seen from above with the webbing raised around it, the vampire squid looks like something assembled from darkness and pointed edges, hence the name. The webbing contains soft filamentous spines called cirri. When the vampire squid pulls its webbed arms up over its head, exposing the inner surfaces of the arms to a threat, it presents a display of white tips on the cirri that some researchers interpret as a confusion tactic, creating visual noise that makes locating the center of the animal difficult for a predator. The vampire squid produces bioluminescence of extraordinary variety and control. Photophores on the mantle and fins can be modulated in intensity and pattern. When threatened, the vampire squid can release a cloud of bioluminescent mucus from the tips of its arms, a sticky, glowing substance that persists in the water for several minutes after the animal has escaped into the darkness. A predator follows the glowing cloud while the vampire squid retreats invisibly in the opposite direction. At the tips of the arms, the vampire squid carries filaments, extremely long retractile threads that extend far beyond the arm tips. These filaments are used for feeding in a way that separates the vampire squid completely from every other cephalopod. It does not hunt. It does not pursue prey. It does not ambush fish or squid or crustaceans. It eats marine snow. The vampire squid extends its long filaments into the water column around it and allows organic particles drifting downward to adhere to them. Sticky mucus on the filament surface captures the particles. The filaments are then retracted and the particles are consumed. The vampire squid is not a predator. It is a marine snow filter, a scavenger of the most degraded organic material in the ocean. It evolved to extract sustenance from the nutritional dust that falls through the oxygen minimum zone where no other significant predator ventures to compete for it. This is a food source of extremely low caloric density. The vampire squid survives on it because its metabolism is extraordinarily slow. Its muscles are weak compared to other cephalopods. Its movement is minimal. It hovers in the dark of the oxygen minimum zone, spending almost no energy. Its lifestyle is the most economical in the cephalopod world. It does less and uses less, and therefore needs less than any related animal. The oxygen minimum zone where Vampiroteuthis infernalis lives is not a static feature of the ocean. It expands and contracts seasonally and over longer climatic cycles. In recent decades, monitoring data suggests the oxygen minimum zones of the world's major oceans are expanding as the ocean absorbs heat from a warming atmosphere. Warmer water holds less dissolved oxygen. The bacteria that deplete oxygen in the zones below become more active. The zone grows. For most of the ocean's inhabitants, this expansion is a catastrophe. Fish leave, crustaceans die. The ecological community of the oxygen minimum zone boundary collapses as the zone itself expands into formerly habitable water. But for Vampiroteuthis infernalis, an expanding oxygen minimum zone is an expanding empire. More habitat, less competition, more marine snow drifting through more of the water column where the vampire squid has evolved to occupy an exclusive ecological niche. The animal that every other species avoids is the one that benefits from conditions other species cannot endure. The deep ocean's most counterintuitive tenant is expanding its territory. There is something else about the vampire squid worth sitting with. It has been in its ecological role for a very long time. The fossil record of Vampiromorphida is sparse, but the cephalopod lineages from which it branched are found in deposits over 200 million years old. The vampire squid is not a new solution to the problem of surviving in the oxygen minimum zone. It is an old solution, a very old one. It found its strategy in the deep water long before the oxygen minimum zone was defined by human oceanography, long before the organisms that share its ocean today had evolved. The darkness of the deep ocean has been its habitat since an era of life on Earth that the surface world has entirely replaced. When you encounter the idea of the vampire squid in the context of its full evolutionary and ecological history, the name Vampiroteuthis infernalis begins to seem less like dramatic scientific naming and more like an honest description. It is ancient. It is from a lineage that the rest of the cephalopod world left behind. It lives where nothing comfortable can survive. And it has been doing this since before the ocean around it looked anything like it looks today. The Siphonophore praia dubia deserves mention in this chapter Because it challenges another boundary entirely, the boundary of the individual organism. Siphonophores are colonial animals. They are not single organisms in the conventional sense. They are colonies of individual organisms called zooids that are so physically integrated that the colony functions as a single animal. Different zooids specialize in different functions. Some produce propulsion. Some handle digestion. Some are responsible for reproduction. Some handle defense. None of the individual zooids can survive outside the colony. None of them constitutes a complete organism on its own. The colony is the organism. Praia dubia forms the longest known organism on this planet. Measured specimens have reached 40 to 50 meters in total length, longer than a blue whale, longer than any known animal. It drifts through the mesopelagic zone, trailing its long structure through the dark water, and trailing also thousands of stinging cells on its trailing tentacles. Anything that makes contact with those tentacles is captured and passed to the digestive zooids for processing. The whole enormous structure hunts as a single organism across a span that would dwarf any individual animal alive. The vampire squid and the siphonophore together demonstrate that the deep ocean contains not only extreme versions of familiar animal categories, but also organisms that challenge the definitions those categories are built on. One is taxonomically alone in the history of its order. The other redefines the word individual. Both are products of a world that operates by rules we are still learning. In the next chapter, we descend to the seafloor and discover what happens at the end of every life that has ever lived in the ocean above. Chapter 11. The bone eaters, zombie worms, whale falls, and the ecology of death. A whale weighs up to 150,000 kilograms. It is the largest animal alive. When it dies, its body falls. This does not happen quickly. A dying whale often floats first. The gases of decomposition keep the carcass near the surface for days or weeks, while seabirds and sharks strip the outer layers. Eventually, the buoyancy fails. The carcass sinks, and as it descends through the water column, it passes through every zone described in this documentary. Through the twilight zone where the viperfish and fangtooth live, through the midnight zone of the anglerfish, down through the darkness and the pressure until it reaches the seafloor at whatever depth the ocean beneath it happens to be. When the whale hits the seafloor, something extraordinary begins. Oceanographers call it a whale fall. And what a whale fall creates in the deep ocean is something that has no equivalent anywhere else in that environment. A sudden, enormous, concentrated deposit of organic material in a place that normally receives only the scattered dust of marine snow. A feast measured in tons, delivered to a seafloor community that normally subsists on almost nothing. The arrival of a whale carcass at the deep sea floor is an ecological event that transforms a patch of otherwise barren sediment into a complex and active community for decades. The sequence has stages. In the first stage, which can last months to over a year, mobile scavengers arrive, giant isopods, rat-tail fish, hagfish, sleeper sharks that move slowly along the seafloor. These organisms consume the soft tissue, the muscles, the organs, the fat. They reduce the whale from a complete carcass to a skeleton relatively quickly in the context of the deep ocean. Relatively quickly in the deep ocean means months, but compared to the bare bones phase that follows, it is a rapid transformation. In the second stage, enrichment opportunists arrive. Polychaete worms in their thousands, crustaceans, small invertebrates. They colonize the sediment around the skeleton that has been enriched by the fluids and breakdown products of the decomposing carcass. They exploit the elevated nutrient levels in a thin zone of seafloor around the whale. This phase can last years. Then comes the third stage, the one that keeps running long after the soft tissue and the enriched sediment are gone. And this is where a group of organisms enters the story that biologists did not know existed until 2002, the bone worms. Osedax. The genus name is Latin for bone devourer. They were discovered by a team of researchers from the Monterey Bay Aquarium Research Institute who observed small worms growing directly from the bones of a decomposing gray whale on the seafloor of Monterey Canyon at approximately 2,900 meters depth. The worms were unlike anything previously known. Their anatomy included structures that made no sense in the context of known annelid worm biology. The researchers collected specimens and spent several years working out what they had found. What they had found was a worm that does not have a mouth. Osedax enters bone not by biting or rasping, but through a different biological mechanism entirely. The worm grows root-like structures into the bone tissue. These roots secrete acid. The acid dissolves the calcium phosphate mineral matrix of the bone. The dissolved material, along with the organic compounds and lipids trapped inside bone tissue, is absorbed directly through the root surfaces and passed to symbiotic bacteria living within the root tissue. The bacteria metabolize the bone lipids and provide nutrients back to the worm. The worm has no conventional digestive system. It does not eat in any conventional sense. It dissolves from the inside out and absorbs the products. The discovery of Osedax forced a revision of assumptions about deep-sea ecology that had been in place since oceanography became a systematic science. Before Osedax was found, the deep-sea floor was understood to be a nutrient-limited environment where the main food inputs were marine snow and the occasional large food fall. The role of bone as a long-term food resource had not been characterized. Researchers assumed bones were processed by bacteria and physical degradation over time. Osedax revealed a highly specialized biological system for bone processing that is more efficient and more rapid than passive decay alone. Subsequent surveys found Osedax in every ocean that was looked at: Atlantic whale falls, Pacific whale falls, Antarctic waters, the Mediterranean. Osedax species were found on fish bones as well as whale bones, on the bones of marine turtles, on the bones of seabirds that sank. The worms are not limited to whale falls. They colonize any bone material that reaches the seafloor at depth. The deep ocean floor is, it turns out, covered in the chemistry of dissolution being performed by worms nobody had ever seen before. 2002. The chemistry of what Osedax does to bone also has broader ecological significance. Whale bones are rich in lipids, fatty compounds stored inside the bone matrix that represent enormous reserves of caloric energy. A large whale skeleton contains thousands of kilograms of bone lipid. Osedax accesses this resource and processes it into forms that enter the deep-sea food web. The carbon and energy locked inside whale bone moves through Osedax into the bacterial symbionts, and then into the worms themselves, and then into the predators and scavengers that feed on the Osedax colonies. The whale fall is not merely a pile of food. It is a slow chemical reactor converting bone into biological currency that the deep-sea ecosystem can spend over the following decades. But the reproductive biology of Osedax is where the story takes a turn that should be familiar by now. The females are the visible worms, centimeters long, growing from the bone surface in dense groups, their feathery red gills extending into the water above the skeleton. The males of most Osedax species are microscopic. They live inside the body of the female, not attached externally as the male serratioid anglerfish attaches to the female, inside her. Within a special structure in her body tube, a female Osedax may contain dozens or hundreds of microscopic males living in her tissues simultaneously. The males exist exclusively to produce sperm. They have no other function and no other life stage. The deep ocean produced independent solutions to the same reproductive problem in two entirely separate lineages. The anglerfish and the bone worm both arrived at a version of male miniaturization and female dependency. The anglerfish fuses male and female through tissue merger. The bone worm houses the males internally. the mathematical pressure behind both solutions is the same. Encounters in the deep ocean are rare. When a male finds a female, he must not leave. A single whale skeleton on the deep sea floor can support an active community of osedacs for decades. Some researchers estimate that a large whale skeleton can sustain boneworm communities and the organisms that feed on them for up to a century before the bone is fully processed. One death, one falling body. 100 years of concentrated life in an environment that normally sustains almost nothing. There is a fourth stage to the whale fall that occurs after the osedacs communities have processed the bone lipids. In certain conditions, the sulfur in the remaining bone material supports communities of chemosynthetic bacteria that use hydrogen sulfide as an energy source rather than sunlight or organic carbon. These sulfur-based bacterial communities in turn support tube worms and clams and other organisms similar to those found at hydrothermal vents. A whale fall at its final stage becomes a miniature chemosynthetic ecosystem, a temporary vent community powered not by geothermal heat, but by the chemical legacy of a single large body. The 4 stages of a whale fall span decades. They represent a complete ecological succession from scavenging to decomposition to bone processing to chemosynthesis. Each stage supports different communities. Each community is replaced by the next as the resource state of the carcass changes, and the whole sequence is powered by a single death falling through thousands of meters of dark water. In an environment as austere as the deep sea floor, a whale fall is a gift of almost unimaginable proportions. The ecosystem builds around it, uses it, transforms it, and keeps going until the last molecule of usable chemistry is extracted. Then, the sediment settles over what remains, the community disperses, and the abyssal plain returns to its normal silence. The whale fall is the most dramatic example of what ecologists call a trophic cascade. A single event at one level of the food web, creating consequences that ripple outward and downward through dozens of other species and across timescales that the surface world would consider geological. In the deep ocean, a death is not an ending. It is an event that begins something new and sustains it for longer than most surface ecosystems can sustain anything at all. In the next chapter, we arrive at the deepest flat surface on the planet, the abyssal plain, where the sediment is ancient and the creatures that cross it are among the strangest things in the ocean. Chapter 12. The Abyssal Plain, the strangest landscape on Earth. Imagine a landscape that covers more than half the surface of this planet. A flat expanse of fine sediment extending without significant interruption in any direction as far as any instrument can detect. No mountains of significant height, no valleys of significant depth. Just a vast level plain of compacted organic debris and mineral particles that has been accumulating for tens of millions of years at the rate of roughly 1 centimeter per 1,000 years. The sediment at the base of the deepest abyssal plains was deposited before the first dinosaurs walked the Earth. This is the abyssal plain, the region of the deep seafloor between approximately 4,000 and 6,000 meters depth. It covers an estimated 300 million square kilometers of the ocean floor. It is the most common terrain on this planet, and for most of recorded human history, we assumed it was essentially lifeless. We were wrong. The abyssal plain is not dense with life in the way a coral reef or a kelp forest is dense with life. The food supply. Degraded marine snow arriving at rates measured in millimeters of accumulation per millennium does not support a high-energy ecosystem. What it supports instead is a low-energy ecosystem of extraordinary diversity and extraordinary strangeness. Organisms that solved the problem of surviving on nearly nothing by becoming nearly nothing themselves, moving almost imperceptibly, feeding on particles smaller than sand grains, living for decades on a caloric budget that would sustain a surface organism for days. The abyssal plain sediment itself is worth describing in more detail because it tells the history of the ocean in a form that can be read by scientists who know how to look at it. The sediment composition varies across the plain. In some regions, it is dominated by calcium carbonate from the shells of foraminifera, microscopic single-celled organisms that live in the upper ocean and whose shells rain down when they die, contributing to the sediment that has been accumulating since the Cretaceous. In other regions, silicate sediment from diatoms and radiolaria dominates. In the deepest regions, a red clay accumulates that is one of the most slowly accumulating materials on the planet, a few millimeters per million years. The clay is so old and so slowly deposited that its upper layers contain material that arrived during the last ice age, and its lower layers contain material deposited before the ancestors of most modern animal groups had evolved. Walking across the abyssal plain would be like walking across a geological library. Every footfall compressing millions of years of biological history into the sediment under your feet. The abyssal plain is not simply a floor, it is a record. Begin with the sea cucumber. The sea cucumber relatives that dominate the abyssal plain belong to the genus Scotoplanes and their relatives. Common names include sea pig. The name is not elegant, but it is accurate. They are pink. They are soft. They stand on enlarged tube feet that project downward and function as legs. In high-definition footage, they move across the sediment surface in groups, sometimes in herds of dozens or hundreds, crossing the abyssal plain on their fleshy legs, processing sediment through their bodies as they go. They eat by taking in sediment at the front end and extracting whatever organic content it contains before expelling the cleaned particles at the rear. Sea pigs are the most biomass-abundant animals on the abyssal plain in many regions. They exist in densities that seem impossible given the food supply, but their metabolism is calibrated to a precision that makes the most of every particle. They represent the largest mobile grazing community in the most extensive habitat on Earth, and they are, by almost any measure of appearance, one of the most bizarre animals on this planet. Now meet an organism that stretches the definition of an animal in a different direction. Xenophyophores, single-celled organisms found on the abyssal plain. Single-celled in the strict biological definition. One cell, one nucleus complex, one cellular boundary. But the cell can reach 20 centimeters in diameter. It is the largest known individual cell of any organism. A single-celled organism the size of a volleyball, visible to the naked eye, visible without any magnification. The xenophyophore accumulates grains of sediment and incorporates them into a structural skeleton called a test that gives it a complex, irregular shape. It processes sediment through its cell body, extracting organic material. It reproduces by fragmenting. The xenophyophore holds another record. It was found alive at 6,300 meters depth in the Mariana Trench. At that depth, a single-celled organism lives in a pressure environment that would destroy most cellular machinery. The xenophyophore apparently manages this through biochemical adaptations that are still being characterized. The abyssal plain also hosts polychaete worms of extraordinary diversity, hundreds of species. Most are not individually dramatic, but their collective function in the abyssal ecosystem is critical. They process sediment. They aerate it by moving through it. They provide prey for the larger mobile scavengers, and some of them produce the most elaborate tube structures of any marine worm. Building homes from sediment particles cemented with secreted mucus, homes that can persist long after the worm that built them is gone and that become habitat for other organisms in turn. The distribution of life on the abyssal plain is also not uniform. Areas directly beneath highly productive surface ocean regions, where plankton blooms and fish populations create large quantities of sinking organic matter, support more life per square meter than areas beneath the central ocean gyres where surface productivity is low. The abyssal ecosystem is tethered to the surface world by gravity, even as it exists in conditions that have nothing in common with the surface world. What falls determines what lives. What lives is shaped entirely by what falls. This connection between the surface and the abyss is one of the most important in global ocean ecology. The carbon cycle, the process by which carbon dioxide is absorbed at the surface and eventually sequestered at the bottom, runs directly through the abyssal plain. Every organism that eats in the deep is part of the mechanism by which the ocean moderates the carbon content of the atmosphere. The abyssal plain is not separate from the world above. It is the bottom of a system that encompasses the entire ocean and influences the chemistry of the air we breathe. Then, there are the sea spiders, Pycnogonida, marine arthropods found in oceans at all depths but reaching their most extreme sizes in the deep and cold ocean. The abyssal and Antarctic sea spiders can have leg spans of up to 70 centimeters. They are not true spiders, they are arthropods in their own separate class. Their bodies are so reduced that most of the digestive organs have been displaced into the legs. They consume prey by inserting a proboscis and sucking out the interior. They move slowly across the seafloor, hunting small invertebrates. The hydrothermal vents that occur at some abyssal locations represent the most extreme contrast to the surrounding sediment. Around volcanic ridges where tectonic plates separate hot mineral-rich water emerges from vents in the seafloor at temperatures that can exceed 350 degrees Celsius. The water is kept liquid by the enormous pressure. Around these vents, communities of organisms that owe nothing to sunlight and nothing to marine snow conduct their entire lives on chemistry derived from hydrogen sulfide and other volcanic compounds. Tube worms, 2 meters tall. Giant clams, white crabs, all built on a foundation of chemosynthetic bacteria. These vent communities were discovered on February 17, 1977, when the deep-sea submersible Alvin was exploring the Galapagos Rift. The researchers on board were expecting to see a barren volcanic landscape. Instead, they found a dense community of living organisms unlike anything previously known. The discovery rewrote the rules about where life was possible and is still considered one of the most significant biological discoveries of the 20th century. The abyssal plain is not a wasteland. It is the most extensive biological community on Earth. It runs on almost nothing and produces almost no heat and makes almost no sound. But across its 300 million square kilometers, something is always eating and something is always being eaten. Slowly, patiently, in the deepest silence in the world. In the next chapter, we go to the very bottom, to the place where even the abyssal plain is far above. Chapter 13. The deepest horror. Hadal zone creatures at the limit of possible life. Below the abyssal plain, the seafloor is interrupted. Not everywhere. Mostly the deep seafloor is indeed the flat sediment plain described in the previous chapter. But at the boundaries of the great tectonic plates where one plate slides beneath another, the ocean floor drops into long, narrow trenches of extraordinary depth. The Mariana Trench, the Tonga Trench, the Philippine Trench, the Japan Trench, the Izu Ogasawara Trench. These geological fractures plunge below the abyssal plain in the same way that canyons plunge below a plateau, and the zones they create. The Hadal Zone, named for Hades, the underworld, represent the most extreme habitable environment on the surface of this planet. 6,000 meters to 11,000 meters depth, pressure ranging from 600 to over 1,100 atmospheres, temperature near freezing and stable. Nearly no light except what organisms generate themselves. Almost no food except what settles into the trench walls and floor from the water column above. For most of scientific history, the question of whether anything lived in the Hadal zone was genuinely open. The pressures seemed too extreme. The food supply seemed too poor. The environment seemed too hostile for any metabolism to function within it. The early deep-sea surveys of the 19th and early 20th centuries went as deep as their equipment allowed and found progressively less life with each additional depth increment. The extrapolation pointed toward a completely lifeless abyss at maximum depth. The extrapolation was wrong. In 2023, a research team deployed a baited camera to the floor of the Izu Ogasawara Trench. The camera descended to 8,336 meters at that depth, in a pressure of over 800 atmospheres. At temperatures just above zero, the camera filmed fish, swimming, active, feeding on the bait, snailfish, Pseudoliparus belyaevi, members of a family of fish found at extreme depths across multiple Pacific trenches. 8,336 meters. That depth is currently the confirmed record for the deepest living fish ever observed. A fish with a recognizable body plan, fins, eyes, normal vertebrate anatomy, living without apparent difficulty in conditions that would destroy the structural integrity of almost any other vertebrate body within seconds. The hadal snailfish, Pseudoliparus belyaevi, and related species are not large. Adults measure between 10 and 30 centimeters. They are pale and soft-bodied, largely translucent in appearance. Their skin is not pigmented in any significant way because at these depths there is no functional color. Their bodies are built around the same gelatinous tissue architecture that the rest of the deep ocean favors. Flexible, pressure-tolerant, without the rigid internal air spaces or large calcified structures that would be destroyed at depth. They are surprisingly normal-looking to eyes accustomed to the dramatic anatomies of the anglerfish and the fangtooth and the goblin shark, and that is itself a piece of information about the hadal zone. At the deepest levels of the ocean, the primary evolutionary challenge is not predation and not competition, it is chemistry. The organisms that live in the hadal trenches are not the most visually extreme creatures in the deep ocean. They are the most biochemically extreme. The most extraordinary thing about them is invisible. It is molecular. It is in the concentration of a stabilizing compound in every cell of every tissue in their bodies. The visible extreme is sometimes less extreme than the molecular one. How does it survive? The answer is a molecule, TMAO, trimethylamine oxide, a chemical compound that cells use to stabilize proteins against the effects of extreme pressure. Proteins are the working machinery of cellular biology. They are three-dimensional structures whose shape determines their function. Pressure at depth distorts protein structure the way squeezing a spring changes its shape. Distorted proteins stop working. A cell full of distorted proteins is a cell that cannot function. A body full of cells that cannot function is a dead body. TMAO accumulates in cells under pressure and physically counteracts the deformation that pressure would otherwise cause. It acts as a molecular shield between the cell machinery and the crushing environment outside. The concentration of TMAO in cells increases predictably with depth. Fish at 1,000 meters carry more TMAO per cell than fish at 200 meters. Fish at 4,000 meters carry more than fish at 2,000. And snailfish at 8,336 meters carry the highest concentrations of TMAO ever measured in a fish. There appears to be a limit to how much TMAO a cell can contain before the concentration itself becomes biologically disruptive. Researchers have estimated based on known TMAO concentrations and their rate of increase with depth that this limit is reached somewhere between 8,000 and 8,500 meters. If this estimate is accurate, the snailfish filmed at 8,336 meters was operating near the absolute biochemical limit of vertebrate life, not simply at an extreme depth, at the maximum depth where vertebrate cellular biology can function at all. The hadal trenches are not only inhabited by fish, the trenches themselves are geologically active environments. Food arrives at trench floors through two pathways: the marine snow pathway, organic particles settling from above, is the same mechanism that feeds the rest of the deep ocean, though the conical geometry of the trench may concentrate particles from a larger surface area of water above down into the narrow floor. The second pathway is turbidity currents. Sediment avalanches, triggered by earthquakes along the trench walls that carry organic-rich sediment from shallower depths down the trench walls and concentrate it on the floor. Subduction zone trenches are associated with active tectonic boundaries. The same boundaries produce the earthquakes that trigger tsunamis and the volcanic activity of the Pacific Ring of Fire. The organisms that live in hadal trenches live not only in the deepest and most pressurized environment on Earth, but in an environment that is geologically the most active: periodic sediment avalanches. Seismic disturbances, the occasional arrival of large organic material carried down from above by tidal and turbidity flows. All of this creates a dynamic that is not simply extremely deep, but extremely unpredictable in its pattern of food delivery. The hadal scavenger community is built for exactly this unpredictability. High-speed arrival at bait, aggressive competitive feeding, the ability to fast for long periods between food events, maximum efficiency of consumption when food is present. Amphipods. Crustaceans in the order Amphipoda are often the most abundant animals in hadal environments. Species like Hirondelia gigas and related forms colonize trench floors in densities that seem impossible given the food supply. They are scavengers. Attracted to food falls from above with the same speed and urgency that gilled shark are attracted to whale carcasses on the abyssal plain. When a bait camera is deployed in a hadal trench, the amphipods typically arrive within minutes, hundreds of them, swarming the bait, competing, consuming, a community of life that was there, waiting, in the dark, before the camera arrived. The hadal amphipods carry TMAO as well. They accumulate it in the same manner as the fish. Their chitin exoskeleton, unlike the bone of fish, is not subject to the same pressure deformation problems. But their cellular machinery faces the same chemical environment. They adapted through the same molecular strategy. Xenophyrophores appear in the hadal zone as well. The enormous single-celled organisms described in the previous chapter were found alive in the Mariana Trench at 6,300 meters. This makes them among the deepest known eukaryotic organisms, single cells surviving at pressures 600 times greater than atmospheric. Their mechanism for pressure tolerance is not yet fully characterized. They are among dozens of hadal species, about which we have only the most basic information. The hadal zone represents the final extreme of the gradient this documentary began with. Darkness, pressure, starvation at their maximum values, and life against all extrapolation. At every depth we have managed to look, life is there. In the final chapter, we come back to the surface and ask the question that all of this was building toward. Why? Why does the deep ocean produce what it produces? And what does it mean for everything we think we know about life? Chapter 14. Why Darkness Makes Monsters. The grand synthesis of deep-sea horror. We began this journey with three forces: darkness, pressure, starvation. And we said those three forces working together over hundreds of millions of years explain everything, every creature we encountered, every adaptation that looked like horror, every feature that seemed designed to disturb. The anglerfish with its borrowed bacterial fire and its cage of transparent teeth, the goblin shark whose jaw fires forward from beneath its ancient snout, the black swallower whose stomach inflated around a meal that outlasted the fish that swallowed it, the male anglerfish dissolving its own body into its mate until two separate organisms become one permanent thing, the boneworm with no mouth that dissolves bone from inside using acid and its own roots. All of them, every one of them, is evolution responding rationally to conditions we have never had to experience. The deep ocean did not produce monsters, it produced solutions. And the solutions look like monsters to us because we evolved in the sunlit world above, and our minds were built by conditions that have almost nothing in common with the midnight zone. But there is a deeper pattern that emerges when you step back from the individual creatures and look at the full arc of what we have described. It is not just that darkness and pressure and starvation produced extreme biology. It is that they produced extreme biology through convergence, separately, independently. In lineages that share no recent common ancestor, the same pressures generated the same solutions again and again across the tree of life. Sexual parasitism, in which the male fuses with the female or lives inside her, appeared in the anglerfish and independently in the bone worm Osedax. Two completely different animal groups, different phyla, different evolutionary histories, different body plans. Both arrived at a version of male miniaturization under the pressure of rare encounters in a food-scarce dark environment. Bioluminescence evolved independently more than 40 separate times in different deep-sea lineages. The deep ocean provided the same conditions everywhere, and those conditions generated the same tools everywhere, regardless of which lineage was doing the evolving. Convergent evolution is one of the most powerful concepts in biology. It tells us that when conditions are extreme enough and consistent enough, they constrain the solution space so tightly that independent lineages arrive at the same answer. The answer is not random. It is forced by the environment. The deep ocean is the most powerful demonstration of convergence on this planet. It is a laboratory that ran for hundreds of millions of years with consistent conditions and consistent pressures. And it produced consistent results across dozens of separate experiments. Consider what convergent evolution in the deep ocean means for our understanding of the history of life specifically. The deep ocean has been chemically and physically stable for far longer than the surface world has been stable. The conditions in the midnight zone today are not significantly different from the conditions in the midnight zone during the Cretaceous. The organisms we find there today are shaped by pressures that were already operating when the continents were in different positions, when mammals were small, when birds had not yet diversified, when the surface world was a completely different place. The deep ocean did not wait for the surface world to change. It ran its experiments continuously, generation after generation of organisms in the same conditions, learning the same lessons. The fangtooth had long teeth in the Cretaceous. The principles that produced bioluminescence were already working in the Jurassic. The convergent solutions we see today are not new. They are the current expressions of very old mathematics, the same equations running on different hardware across different geological eras, arriving at the same output each time because the conditions demanded it. What does this tell us about life more broadly? It tells us that life is less creative than we sometimes imagine and more predictable than we sometimes fear. Where conditions are sufficiently extreme, the range of viable solutions shrinks. Bioluminescence in darkness, elastic stomachs in food-scarce environments, giant eyes in dim light, pressure-tolerant biochemistry at depth. These are not surprising inventions. They are mathematically required responses to the variables involved. Given enough time and the right conditions, life will find them. Every time. This has implications that extend beyond the deep ocean. In the search for life elsewhere in the universe, the deep ocean of Earth has become one of the most important reference points. Europa, one of the moons of Jupiter, is believed to have a liquid water ocean beneath its ice surface, estimated at between 60 and 170 kilometers deep. Enceladus, a moon of Saturn, ejects plumes of water and organic compounds from beneath its south polar surface. Both bodies appear to have the conditions that the deep ocean of Earth demonstrates are sufficient for life. Liquid water, chemical energy sources, stable conditions over geological time. If the lesson of the deep ocean is that life finds solutions wherever the conditions allow, then the existence of life on Europa or Enceladus is not a remote possibility. It is a rational expectation. We do not know what it would look like. We do not know whether convergence would produce recognizable analogues to the anglerfish or the vampire squid or the boneworm, but we know that the pressures that produce extreme biology on Earth are not unique to Earth. And we know that extreme biology exists wherever the pressures are sufficient to produce it. The rate of discovery in deep-sea biology has accelerated dramatically in recent decades. In the 1970s, the only way to sample the deep ocean was with trawl nets that destroyed everything they collected. The carcasses that arrived at the surface told us what lived at depth, but told us almost nothing about how it lived. The introduction of remotely operated vehicles and autonomous underwater vehicles with high-definition cameras changed this. For the first time, it became possible to observe deep-sea organisms in their natural state, moving, behaving, interacting, and every time a new vehicle reached a depth that had not been surveyed before, it found organisms that had not been described. The discovery rate has not slowed. Every major deep-sea expedition returns with species new to science, not new variants of known species, new species entirely, animals belonging to groups that were not known to exist in the environments where they are found. The census of marine life completed in 2010 estimated that the deep ocean probably contains millions of undescribed species. The actual number is unknown and cannot be estimated with confidence, because we simply have not looked. The deep ocean is not a solved problem. It is an open one, the largest open question in biology about the living world on this planet, and it is located under the water that covers most of the surface of the world we live on. There is one final thing worth saying about the deep ocean as this journey ends. We have seen less than 20% of the deep ocean floor in any meaningful resolution. Less than that has been surveyed with the instruments capable of detecting biological life at fine scales. The creatures described in this documentary were discovered in pieces. The giant squid, known only from whale stomachs for most of recorded history. Osedax, found by accident in 2002. The barreleye eye rotation confirmed only in 2009. The ultra-black fish measured in 2020. Each one a discovery that changed the boundaries of what we thought the deep contained. The creatures we have not yet found are out there, in the dark, right now, at depths we have not surveyed with the instruments they would require us to use to find them. In a world that covers nearly a third of the surface of this planet, and that we have only barely begun to look at, whatever they are, whatever they do, whatever solutions they evolved to the same three forces that built everything described in this documentary. They are there: darkness, pressure, starvation. The deep ocean keeps its secrets at the bottom of the world, in a cold that has not changed in millions of years, in a dark that existed before almost anything alive today was born. And in that darkness, the most extreme and the most extraordinary biology on this planet continues doing exactly what it has always done, surviving, quietly, waiting for the next meal. We hope this journey through the deep ocean was something worth taking. If you enjoyed it, please consider leaving a like and subscribing to Earth at Night. Your comments below mean a great deal. Share what surprised you most and see what others found when they listened to the same darkness. Good night.